So APP diagnostic, the first 3 points are the major reasons to do that. The most important one, to find the reason for acute clinical symptoms or sudden death cases, but also 2, to find out the serotype which is responsible for the disease, to select appropriate vaccine because you have mainly no cross protection between the serotypes. And 3, if you have a negative farm, you should test pigs which are introduced to the farm if they are also negative for APP. These are mainly gilts. And the next 3 points are not so important. In my opinion, it's difficult to answer them with a diagnostic. For example, find the reason for pathological lung slaughterhouse findings, because very seldom you can culture APP from the lung alterations found in lungs from slaughterhouses, because it grows very delicate and mostly it's not possible. And if you want to test for the protectivity of vaccine, of course, it's not a proof if you find the pathogen, because you will not have a sterile immunity after vaccination. And at last, the point 6, some practitioners ask for getting an epidemiological link. So they want to find out the origin of the APP strain in their herd and if it might have been introduced from another farm. In my experience, it's very difficult because you have to do whole genome sequencing and biostatistics, and this is very cost and personal intensive. And we can discuss it later, but I had no case where this has been successful in the past. So this is a routine diagnostical procedure. Most important, the culture shown on the left, and APP is mainly biotype 1, which is NAD dependent, and the typical growth you can see here on the plate with the colonies growing tightly in neighborhood to the nurse strain, which is producing APP, in this case is Staphylococcus strain. And if you have a pure culture, you can type the capsular genes by PCR. This is, in my opinion, the gold standard. In addition, you can do typing of APX toxin profiles, so you get groups of APP, and you can further specify by agglutination with rabbit antisera, but you will only get the serotyping groups, but you have the chance to detect also APP with atypical toxin patterns. You can do PCR directly from lung tissue, but mainly or mostly you do not have the possibility to find out the serotype which is involved. This is only possible if you have very large burden of pathogen. And serology is used to test if pigs are free of APP. So APX4 ELISA is very common because it's only positive if the pigs has been infected, not if they has been vaccinated. You can do the LPS ELISA, and you can do also serotype-specific ELISA. Then you get already groups, and results are difficult to interpret sometimes because the pig can have contact with several serotypes, and it's hard to find out the one which is most important in the farm. So according to the first 2 points, necropsy and bacteriological examination is most important. And here you can see pictures from experimental monoinfections with APP, and you see the effects of the APX toxins, which are the most important virulence factors, very clearly. Already 7 hours after infection, you can see these small red dots. You can imagine that single bacterial cells or cell clusters in the lung tissue produce APX toxins immediately, and the alterations will become confluent around 48 hours after infection, and you see the typical picture of porcine pleuropneumonia. And later on, when the pigs do not show clinical symptoms anymore, when they are chronically infected, here on day 21, you have these lung lesion sequesters or abscesses in the lung. If you want to test if pigs are negative for APP, we recommend to combine serology and tonsillar sampling, followed by PCR. At best, with the method here shown on the right side, tonsillar brushing, because it's invasive enough to get material from the crypts, but it's not so harmful as tonsillar scraping, as shown here in the middle. Of course, you have no information about the clinical impact of the tonsillar APP strains, But in our opinion, it's important to add it to serology because often pigs which are only colonized with APP in the upper respiratory tract have no detectable antibodies. So it's always good to be safer to use also PCR. And it's also good to know the infection dynamics on your farm because then you can define the best time point for diagnostic and you can elaborate control measures. At the very first beginning, you might have had an introduction of APP by asymptomatic carrier pigs, then a spread in pig groups with sometimes high morbidity and variable lethality until the pigs develop immunity and became subclinical carriers. And then you have this endemic infection. And an important point is the transmission of the agent from the sow to their piglets, so that the piglets become colonized around day 10 of life. And if you have maternal antibodies against APP, this colonization can be prevented or postponed, because when the pigs are mixed in nursery, the infection is spread during the decrease of maternal antibodies around week 6 of life, so that most frequently you get outbreaks with high morbidity in week 12 of life. So to control a free farm is, of course, to avoid introduction of APP into the herd by external biosecurity and continuous monitoring. But most important is control in endemically infected farms, as we have heard in our first poll. And this is difficult. If you implement measures for control, you have to check, do and check again, because it can change over time, the efficiency of your methods. And a very big challenge is to reduce the direct contact between pigs, which is very important. Up to now, it's not known why some sows transmit the pathogen to their piglets and others not, although all sows might be positive in an endemically infected herd. And within the litter, some studies shows that the litters can be affected very variable, ranging from 8% to 100% of the piglets, but also up to 40% of the litters can be negative until weaning. And up to now, we do not know what happened during this transmission process from sows to piglets. But control means limit cross-fostering already in the suckling period, do not mix pigs, and it's always good to reduce the number of pigs per pen. And here I show you a model from human medicine about the development of respiratory disease. And in my opinion, this might be also true for APP. In a healthy host, you have a homeostasis, which means a neutral dispersion model with an intact microbiota in the tonsils, and continuously bacteria are translocated in lower amounts but in a high diversity to the lung and eliminated here by the cells. But if you have any trigger or cofactor, which can be a sudden one as a viral infection or a more long-lasting one like stress, the microbiota in the tonsil is disturbed, and especially pathogenic microorganisms as APP can proliferate, and you have a change in growth conditions so that they might have a growth advantage. You have immune reactions, and a large amount of the same species, so a low diversity, can be translocated to the lung and cannot be eliminated, and you will have disease. This can be exaggerated by dysbiosis in the gut, which might have been triggered by antibiotic treatment. And in worst case, you have a translocation from gut bacteria to the lung. So this means that control has to take into account the trigger factors, and there are several of them. In my opinion, climate stressors are very important, although there are no scientific reports about them. But if you ask farmers and practitioners, they always say that they have an impact. So it's more empirical observation. But if you go to a farm where outbreaks take place and you look for these factors, you will mostly find them, as in these pictures. Here we have false airflow. On the right side, you have an outer climate stable and drought in the lying area. you have leakage between ceiling and wall. In this case, you see the water running down and the pigs feel very uncomfortable. And on the right picture, this was an outbreak in sows. The sows died suddenly in the breeding unit. And when I knelt down to the sleds, I felt the air drought coming from below the sleds. So we find these factors, but we do not know their real impact on disease pathogenesis. But my recommendation is control the air drought and, of course, other abiotic stressors. Other additional factors might be the coinciding pathogens. And I evaluated pigs we had in our diagnostic lab, which were positive for APP and showed the typical picture. And we looked for other pathogens in these pigs. And we compared our findings with this article from Sade, who described or made a meta-analysis, a review article about co-infections and pathomechanisms in the porcine respiratory tract and what has been proven so far to be really synergistic or additive. And they found or summarized that influenza virus has a very high impact, also porcine circovirus 2 and Mycoplasma pneumoniae. And in my opinion, if you have influenza virus in addition to APP, you get very detrimental disease, so very severe disease. So it's a very important secondary or very important coinciding pathogen. There are contradictory findings concerning PRRS virus. So some scientists say yes, it has an additive effect, but in other studies it could not be shown. In my opinion, it can be of high importance. And the differences might be due to the differences in the virus strains, in the PRRS virus strains. And at last, Streptococcus suis, we found in vitro some synergistic effects. I will show you one, and I'm convinced that this agent can also play a role in pathogenesis. But my first recommendation would be control the influenza virus infection. Coming to the host, of course, we have various factors which influence disease pathogenesis. We know that the most important immune cell in defense are the neutrophils, so the innate immunity. Here you see a bronchus of an infected pig with several neutrophils inside. And the neutrophils have important defense mechanisms. You know that they can phagocyte, but they can also produce nets, among other things. And the nets you can see on these immunofluorescent sites. Nets are produced to catch the bacteria. They are consisting of DNA histone complexes, which are shown in green here, and in blue are the neutrophils. And on the left hand, you see the negative control, in the middle, the positive control, and on the right, you can see neutrophils which were in contact with APP, and you can see that APP is a very great net trigger. And this is also shown in this bar graph, in the orange bar. More than 80% of the neutrophils produce nets when they are in contact with APP. But APP is not killed by the nets, because these nets can be cleaved by DNAs from the host or by DNAs produced by Streptococcus suis. And so finally, APP can have a growth advantage by these degraded nets because they are a source for NID. And in our opinion, it might be important also to control Streptococcus suis on some farms, also with respect to APP. And one slide, according to phagocytosis, it's difficult to measure it, but in some labs, a survival factor can be determined, which means that you take serum from some pigs of your farm and mix the serum with washed neutrophils and add the bacterium, and you look if the bacteria can proliferate during 2 hours, and you make the quotient between the amount of bacteria you started with at the beginning and how high is the bacterial load after 2 hours. And it should be below 1, so the bacteria should not proliferate. Then you can assume that opsonophagocytotic antibodies are in the serum. And in this farm, they had very big problems by APP serotype 6, and you see that we sampled several age groups. All the pigs had been vaccinated for a long time with different vaccines, but there were always recurrent outbreaks. And I just want to show you the results of the APX4 ELISA on the x-axis with the cutoff at 50, shown here, and the results of the survival factor measurement. And you see that there's no correlation between these routine assay and the functional test. So you have a low number of animals where the bacteria could not proliferate, here at 0, but you have a high number of animals which are negative in the APX4 ELISA and not protected by opsonophagocytotic antibodies, but you have also the opposite. So there's no correlation, and this is a big problem we have in the moment, that we cannot predict if pigs are protected by no test. So we have to use vaccines against APP, and commercial vaccines are available, and some provide cross-protection for all serotypes. Research for new vaccines is ongoing, so this is very promising. And on some farms also autogenous vaccines are used, but their success is highly variable. Specifically, this might be due to levels of this protein, PALA, which has been found to counteract protective antibodies against APX toxins. So, and these antibodies we want to have because they would be protective. But there might be other effects specific to the farms why autogenous vaccines might not help. And I will end with this statement from Sebastian Bunker. It was from the 19th, when only one commercial vaccine was on the market. It was against APP serotype 2, a killed vaccine. And he has a lot of experience also with autogenous vaccines. And he said that the vaccine can be considered as successful when the morbidity is reduced by 20 to 40% and the mortality by 60 to 80%. So in my opinion, it is a little bit disappointing, but on the other hand, it shows us that we have to do more than to vaccinate. Vaccination has to be accompanied by other measures. So I want to summarize. The innate immunity is decisive for disease control. Colostrum antibodies can postpone the colonization of piglets. Don't mix pigs, very important. And trigger factors and co-infections can lead to disease outbreaks, and all vaccination strategies should be accompanied by other measures against the trigger factors and the co-infections.